So I'm going to talk, you heard us talking in the panel a little bit earlier on, and I'm just going to add to what I said earlier about physiology and the importance of physiology. And as I said, we have a campaign now because the science life depends on, that's physiology. And so we have, as you saw, several journals that are basically related to the sections in the American Physiological Society, renal, cardiovascular, neuroscience, etc., etc. And what we're becoming to know and to understand is the interaction between all of these disciplines is critically important. And one of our latest journals, Comprehensive Physiology, is now subtitled The Journal of Inter-Organ Interaction, just to emphasize that. And so physiology obviously is something that you're all using at this moment, and I pointed this out in terms of the urine, the drinking, etc., etc., the alcohol, and one of the other things that's important that if you want to get into the common literature is to talk about coffee. You know, is coffee good for you? Is it bad for you? Is caffeinated good for you or bad for you? And again, we have this little joke that if you want to publish in a top journal, you do a survey on caffeine, and it doesn't matter what conclusion you come to, it'll be published. So we advance global health and we're using S2O publishing. And this actually sounds very easy to do bench to bedside. But in fact, as I tried to say earlier on, it's really, really hard. We have, just to give a personal example, we made a discovery, an unexpected discovery about maybe 7 or 8 years ago, formed a little company, and then it folded. Well, it didn't really fold, it's on ice because we couldn't get funding to go to the next level of clinical trials. It was an unexpected discovery, went off in a separate direction, but that was 8 years ago. So maybe in 10 or 20 years, you'll see a new drug come into the market for acute kidney injury, we don't know. So it can take decades, that needs to be archived. We need to be able to find this. And I want to give you just a couple of examples of something that you may not, or you may know, or you may not know. And this comes back to Ritu's point about significance. It's important that journals publish things that are not immediately seen as being significant. When I do peer review for grants and for papers as well, they say, oh, what about the significance? I could not have told you 20 or 30 years ago that looking at flower petal color would have led to siRNA medicines, small inhibitory RNAs that knock out genes and that can, actually there are several companies now in the Boston area and beyond that actually have siRNA therapies for various diseases, including amyloidosis and some others also. And this was based on the observation that when you cross certain flowers, the petal color was not obvious from the genetics of the cross. And it turned out that siRNA, small inhibitory RNAs, were blocking some of the pigments that actually caused changes in flower color. Bacterial defense against invading phages. How does a bacteria prevent itself? A bacteria lives there, a little phage comes, injects its RNA or DNA into the bacteria and kills it. But they've developed a protective mechanism against this, and that protective mechanism led to CRISPR-Cas9, which we've all heard about as being genetic scissors and the revolution of the future. It's already providing hope for patients, at least with circulating cell cancers and cancers of the blood cells. I mentioned in the talk the Gila monster toxin analysis for GLP-1 agonists. You know, some people in this room might be taking these GLP-1s. The reason you're doing this is because it was discovered in the Gila monster venom, in their salivary venom. And why, I'll just tell you a little bit in 1 second why. So the agonist structure was known for a while from mammals, but it kept getting degraded. When it was injected into the bloodstream, it got chopped up and it was ineffective. And therefore, the Gila monster was not chopped up. And that was the model for this. DNA replication enzymes from hot spring bacteria in Yellowstone National Park. Bacteria were harvested, enzymes involved in DNA reproduction were harvested, and the PCR technique, the polymerized chain reaction technique, was created to analyze gene expression. So we need consistent access to this record. We need trustworthy long-lived outlets. Wearable technologies, cystic fibrosis, organ transplantation, GLP-1 therapies have all developed over the long term by these techniques. I told you that pregnancy shapes a lifetime of health. So what you do in pregnancy, of course, people tell me it's always the mother's fault. The mouth may hold clues to brain health, that's for sure. Pig organs can save human lives. So in my own institution, we did the first, I say we, one of my colleagues, Leo Riella, did the first pig organ transplantation. And there's a patient alive now who's had a pig organ for over 9 months, previously needed dialysis. It's not as easy as you think to take a pig organ and stick it into a human. Pigs have huge numbers of proteins that cause gross antigenic reactions in all of us. So they have to be genetically ablated. So these pigs, people are going to, in the future, walk around with a little pig on a leash. And that's my pig. That'll do, pig, I suppose. So this is a fantastic breakthrough. And then what fat genes reveal about obesity is helping to develop and generate cures for obesity. So ending by saying again, watch the full-length video of all of this, and you'll see what we're all about in physiology and S2O publishing. So thank you.